Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 172nd seminar, and I call it on National Science Foundation TIP Directorate Day. Since August 26, 2021, I mean 4 years ago, when the first SynBYSS webinar started with Professor Francis Arnold, a Nobel laureate, Teresa Good, the NSF Bio MCB director, India, the EBRC director, the Biomate CEO, and also Albert, a now tenured professor at NC State University, the SynBYSS speaker community has grown with a total of 450 speakers and more than 2,000 audience. The pioneer speakers include a Nobel laureate, 26 National Academy members, 44 program directors of almost all US funding agencies and also some EU funding agencies, 11 editors of Nature and Cell journals, and also 20 editors-in-chief. We have now 212 rising star speakers, many of whom have received tenure or secured a faculty position. My YouTube channel has also attracted more than 2 million views combined, reaching out to the global community. Celebrating this passionate community members' collective success to form an ever-increasing community, I sincerely thank all SynBYSS people for your enthusiastic support and participation. I will continue to build a diverse and also collaborative community with you probably another 4 more years from now on. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Waleed Nasser. He is a program director at National Science Foundation, Emerging Technologies, Biotechnology, the TIP directorate. That's a new one. I met him last year during the Engineering Biology Research Consortium Council meeting, and he kindly accepted my invite to speak. He is one of the 44 funding agency directors and among 8 NSF directors. I especially thank NSF for being the funding agency with the largest number of pioneer speakers. That means, NSF care about this field and also young people. Warwick, thank you so much for your shaping and supporting the research community, and the virtual podium is all yours now, and thank you again. You're mute.
Well, let me repeat that. Well, thank you, Taisuk. Let me share. I'm sharing my slides at the moment. Thank you for the kind introduction. Good morning, good afternoon, everyone, wherever you are. My name is Waleed Nasser. I'm a program director at the National Science Foundation in the Directorate for Technology, Innovation, and Partnerships, or TIP. And it's my honor really to present to you today and talk a little bit about my directorate, about my program, and also about future things we're thinking about. So let me start by introducing the TIP directorate. It's still a new directorate, about 3 years old now. I think we celebrated 3 years old just a couple of months ago. And TIP, what it does mostly is advances USA competitiveness and social impact by nurturing partnerships and drive acceleration. So we're focused on 3 pillars: really diverse innovation ecosystem, building partnerships between industry, academia, and government. We are mainly focused on technology and translational research, use-inspired application, if you would like to define it in a different way. And we are committed for workforce development, what I would like to call translational talent. I want the new generation of translational talent to take their application, their research from the lab to the market. So TIP has 10 key technology focus areas, and this was assigned to us by Congress, from artificial intelligence to computing to semiconductors, robotics, and multiple other things. And one of the things that also was assigned to us by Congress is the biotechnology, medical technology, genomics, and synthetic biology. And that is my program. I'm a program director for biotechnology at the emerging technology section at TIP. So when we started this endeavor, we wanted to really advance competitiveness and growing the U.S. bioeconomy. And in my section, key TA7 or key technology area number 7, which is biotechnology, medical technology, genomics, and synthetic biology, we decided to focus at the beginning on synthetic biology. And the reason is we identified ecosystems that are ripe for acceleration, followed decades of US government investment, especially also from NSF as well. So two ecosystem we identified was AI ML protein design, and the other one was cell-free systems. I'm going to talk a little bit more about these programs. And in these programs, we tried really to focus on the tip pillars, the one I mentioned, build partnerships between academia and industry. And for that reason, we used Ideas Lab instrument. It's a way to bring researchers from academia and industry, let them brainstorm ideas, let them come up with projects, and then eventually they will get funded through the Ideas Lab instrument. As I mentioned, the focus was on use-inspired application. Basic research, foundational research is fantastic. NSF and other directorates are still committed to that endeavor. But at TIP, we are focused on taking those innovations to the market. And of course, we want to train new translational talent. And it's heartwarming through my interactions with multiple, including Taisuk and others, to see the future of the US biotechnology. And it's bright and it's fantastic, especially when it comes to synthetic biology. And this is really directly from the solicitation. We want to enable new applications of importance to the US bioeconomy. And we would like to consider biosafety, biosecurity aspects, and techno-economic analysis. It's an important way to address, if you want to take your innovation to the market, to have a proper techno-economic analysis. And for that reason, we encourage a lot of folks to do that. There are a rising number of faculty members that are focused on TEA or techno-economic analysis, and I would encourage you to really consult with them as well. So let me start with the first program that we came out of my program, which is User-Inspired Acceleration of Protein Design, NSF USPRD. We just announced the awards. I'm sorry, I'm having issues with my. Here we go. Sorry, again, I just had issue with my presentation here. So again, the focus was to accelerate the commercialization of protein design, enable new applications of importance to the US bioeconomy. As I mentioned, we want to bring together the creativity of academia and industry leaders with their pulse on the economy that can drive the translation of application beyond human therapeutics. Again, we want to focus something on beyond human therapeutics simply because there are other government agencies that are focused in that field. And we want to extend AI ML-based design to enzymes space that is ripe for acceleration. So we announced, and you can see the announcement here. This is a link to the announcement of the 5 awardees from the NSF USPRD. Let me just focus on that. So the USPRD portfolio supports broad efforts to open new pathways for biomanufacturing, and reduce cost in support of the bioeconomy. We had 5 great proposals that were awarded. They're all focused on the US bioeconomy. You can actually look into the announcement that came from NSF to read more about these programs. As I mentioned, these programs brought academia and industry together, and even nonprofit as well. And a lot of these programs were actually a large collaboration between industry and academia. Three of the five are led by industry and two are led by academia. And you can tell there is a lot of academic and industry partners in each and every one of those projects. The second program that came out from the biotech section at TIP was advancing cell-free system towards increased range of application of use-inspired application, or C-FIRE. Again, the goal is to create an alternative approach to biomanufacturing that uses cell-free technologies and enable new applications. There are great advantages to cell-free systems, including rapid turnaround, distributed and retargeted manufacturing, synthesis of cytotoxic substances, and precise control over manufacturing environments. However, the technology is expensive with limited use and limited range of application. So we decided to bring, again, industry and academia together to address current limitation of cell-free system to accelerate adoption, foster a virtuous cycle of learning and investment, and drive the translation of a wide range of applications. These awards are expected to be announced, if not this week, probably next week. And I'll forward that announcement to Taisuk, and maybe he can forward it to you all and enjoy it as well. So with that, let me just this. So with that, I want to thank you all. I have my contact information here. And as I mentioned, we are focused in biotechnology, specifically now in synthetic biology. And we are looking for ripe ecosystems that are ready for acceleration. So if you have any ideas that NSF should invest in, any thoughts on these programs, or any idea that you might have for the future, please reach out to me. My email is wnasser@nsf.gov, and I look forward to hearing from every one of you. And with that, I'll get it back to Taisuk. Taisuk, again, thank you for the invitation, and thank you for giving me the opportunity to talk about NSF TIP as well as my programs.
Thank you so much. It's my great pleasure and honor to have you today. And then I completely, agree with you. After, 25 years of synthetic biology, the field, I believe this is the time to deliver real product rather than, still fundamental, study is very important, but now it's time to deliver something to the public in that way benefit the entire bioeconomy. And I believe that's our mission to do. So thank
Yeah,
you so much.
Taisuk, anyone that would like to reach out to me with ideas, with concepts, I'm really open, and I would like to maybe set up a call with you guys. If you need anything else, I'll be more than happy to answer your questions or address any of your concerns.
Thank you
Thank you,
so much. Yeah, thank you
and
so much. Yeah. So
thank
that's wonderful to hear and absolutely, pleasure to have you today. Okay, so now the main speaker of today with a longer introduction. Dr. Siliang Li earned his bachelor's degree from Wuhan University, China, where he studied virus host interactions and developed antiviral drugs. He recently completed his PhD at Rice University under the mentorship of Caroline Ajo Franklin, where he pivoted into synthetic biology and bioelectronics. So congratulations again. His research has been highly interdisciplinary, integrating concepts from microbiology, systems biology, electrochemistry, material and electrical engineering to modulate the electron transfer processes in single microbes and microbial communities. For, of course, the applications in environmental and health monitoring. He is currently a postdoctoral researcher at Rice, working in collaboration with the Bayer College of Medicine to advance bioelectronic applications for disease diagnosis and treatment. His pioneering work has been recognized by the NSF, the same, NSF, of course, funding agency, research traineeship fellow, and Rice University's George J. Schroeder Jr. Award for Outstanding PhD Thesis. Looking ahead, he will continue to foster interdisciplinary and collaborative research to drive innovations in synthetic and fundamental biology. I listened to his talk in the past, one of the conference, and although he was at the time a graduate student, I believe, he gave an excellent talk. Siliang, thank you so much for your time today, and please take it away. It's all yours now. Thank you.
Okay. Can you see my screen? Okay.
Yes.
Okay, cool. Thank you so much for the introduction, and thank you, Tisak, for your continued supporting of our young generations. And it's really my honor to present on this seminar series. So today I'm going to talk about how we engineer microbial bio sensors for environmental and health monitoring. So the growing population and urbanization have caused a serious environmental and health crisis. We have pollution across different landscape and drugs that contaminate our food chain. And consequently, these toxic chemicals harm our human health and leading to diseases and even death. And according to a study in 2022, the chemical exposure and pollution are among the major factors of death, and which cause nearly 9 million premature deaths per year. Well, there are many things we have to do to address these issues, but to begin with, we have to know what type of chemicals are there in the environment that we are dealing with. And for this, we need low-cost and fast sensing techniques to monitor complex chemicals in different environments. Well, perhaps the best sensor so far exists in nature is the living cells, including all of those different microorganisms. So microbes have involved diverse cellular machineries to sense and respond to their environment, and which we can leverage to engineer biosensors. But the question is that how can we get the information out of the cells? So traditionally, researchers may use a fluorescence protein or luminescence as the reporter of those engineered cells. But in my opinion, um, the most efficient and direct way to transmit information is through electronics. This is because electron transfer is fast, it's continuous, and it works in many different environments. And by blending microbes and electronics, we can create a new class of sensors, which is microbial bioelectronic sensors. So these bioelectronic hybrid systems can be made very small and can be integrated into these devices and deployed into different environment. And for example, we can use those sensors in the waterways, and we can even use those sensors within or on our human body. And we can also implement some Bluetooth components into those devices so that we can receive those informations in a wireless way. And blending living cells and electronics is possible because cells are inherently electrical and can interface with the electronic devices. So for all of the living organisms, including ourselves, are powered by electron transfer. So for our human being, we consume a variety of different food, and we use oxygen as the terminal electron acceptor to generate energy. But for some of other microorganisms, they have evolved different ways than us for metabolism. And some of them live in an environment where there's no energy or where there's no oxygen, and they can use some insoluble substrate as the terminal electron acceptors. For example, metal ion. And in the lab, we can even provide those microbes with an electrode so that the microbes can use electrode as the terminal electron acceptor to generate energy. And this is the bioelectrochemical system that we use to cultivate those microbes. It has an anode and cathode chamber, and we inoculate bacterial cells in the anode chamber so that the cells can use the electrode as the terminal electron acceptor, and the electron will transfer from the anode chamber to the cathode chamber. And between these two chambers, we can put some external load. For example, we can use a potentiostat to control the electron flow. And we can also put some electronic devices between the anode and cathode and use the electricity generated from the system to power those electronic devices. And the bottom is a picture taken from our lab showing you the picture of the bioelectric system in the lab. And in this video, you can see that we can even use the electrons from the microbes to power small electronic devices such as a LED bulb. And this was a project we were designed for high school students to educate them for electrochemistry. And the process by which microbes use insoluble electron acceptors is called extracellular electron transfer, or EET. So this bioelectrical interface has been already rewired for many different field applications. For example, we can use those electroactive microbes for bioremediations to degrade the organic waste from the wastewater, and then use the electricity generated from those microbes to produce bioelectricity. And this type of technique has already been commercialized. And for example, a company called AquaPsych is such a company that used the concept of EET to purify the wastewater on site, and then use the electricity generated from the microbes to offset the energy consumption of the system. And more relevant to the topic today, we can also engineer the EET pathway to be switchable in response to specific environmental analytes for bioelectronic sensing. And we can use synthetic biology to engineer these cells so that in the absence of a target molecules, the EET pathway is off, and the microbes produce a low current. Whereas in the presence of a specific analyte, they flip into a EET on state so that they can generate a high current. And this on-off activity of EET will inform the presence of a specific analyte in the environment. And today I'm going to show you two stories about my research, how I construct bioelectronic sensors. And the first story will focus on selective bioelectronic sensing. And the second one, I will talk about a platform that enables us to develop bioelectronic sensors in a modular way. And in the first story, I'm going to talk about how I use a single receptor, an oxidoreductase, to sense multiple molecules and generate multiple signals. So in the field of biosensing, there is a stereotype that emphasizes sensor specificity. So researchers seek to use one receptors to sense one specific analyte to generate a specific signals, for example, antigen and antibody bonding. But the limitation of this approach is that we have to engineer a new receptors every time if we want to sense a new analyte, and this process is very tedious. And there's a transition in this field from specific to selective sensing, where we can use one receptors to sense multiple analytes and generate a fingerprint for each analyte. So this cross reactivity is just like our own olfaction or our taste. So some people call these selective sensors chemical tongue or nose that can generate a unique signal pattern, which can be tied back to the composition of the sample. So the advantage of this selective sensor is that we can use a fewer sensor elements to sense a much large amount of analytes with a fewer engineering process. And we recently discovered an EET pathway in lactate platypus pentera that we can leverage for selective sensing. Alpentera is a gram-positive probiotic bacteria that is widely used for fermented food. And if you go to pharmacy and then buy a probiotic supplement, you will probably find Alpentera as one of the species in this pill. And we found that a new quinone-mediated EET pathway in alpentera. So on the schematic at the left hand, you can see that this pathway has a NADH quinol oxidoreductase or NDH2. So the NDH2 is located in the cytoplasmic membrane, and it can oxidize the intercellular NADH and then transfer the electrons to the flavin adenine dinucleotide cofactors. And if we have some quinones, for example, DHNA shown you here, present in the environment, the quinone will pick up the electrons from the FAD and carry the electrons through the rest of the EET pathway and then reduce the extracellular electron acceptance. Well, alpenterone cannot synthesize those quinones by themselves, so we have to supply those quinones exogenously for alpenterone to perform EET. And also, I wish to mention that the mechanism is definitely more complicated than what I'm showing you here. There are many things that we haven't figured out. But the take-home message here is that the NDH2 is a key enzyme that is indispensable in this EET pathway. So if we knock out the NDH2 from this pathway and we measure the current density in the bioelectrochemical system, we can see a large drop in the current density. So which means that NDH2 is responsible for the current generation we observe. And we also found that this pathway can quantitatively utilize quinone for EET. And in this data, we normalize the current density to the single cells, and we provide the cells with different concentrations of DHNA. We can see that the cells generated different levels of current, which is proportional to the DHNA, the quinone concentration. And if we plot the current density per cell as a function of the DHNA concentration, we can see that the data can be fitted into the Michaelis-Menten equation, indicating that the EET activity can be described by enzyme substrate kinetics. So the reason why we can use this pathway for selective bioelectronic sensing is that the quinone binding site of NDH2 is promiscuous. This is the AlphaFold predicted NDH2 structure of alpenterone. So it has a N-terminal domain that is conserved, shown you in the blue color, and a C-terminal domain that is embedded in the membrane that is less conserved. And if we superimpose the N-terminal conserved domain from 1 to 409 amino acids to other NDH2 homologs that has the crystal structure, and we can see that they share a very similar structure. And at the top is the predicted NDH2 structure of alpenterone superimposed with NDH2 from another gram-positive bacteria, Caldalcalibacillus thermarum. And at the bottom is the NDH2 of alpenterone superimposed with NDI1 from yeast. And it is known that all of these NDH2 homologs can bind to different quinones. Some of them can bind to ladolin, which is vitamin K3, and others can bind to coenzyme Q10. And if I analyze the quinone binding pockets of the NDH2 from alpenterone, I can find a very similar quinone binding domain from the NDH2 of alpenterone. So which means that alpenterone might also be able to utilize different quinones for EET, just like other NDH2 homologs. And to test this, we selected 6 different quinones that share a very similar structure to DHNA. So some of those quinones are naturally found in nature. For example, DHNA, lawson, and juglone, they are all already exist in the nature. And others are synthesized. And some of those other quinones are also manufactured as drugs for disease treatment, for example, anti-cancer drugs. And we supplied those different quinones to alpenterone and compared the current density between the NDH2 wild type versus the NDH2 knockout strains. And we observed that alpenterone generates different electrical signals in response to these different quinones. And by looking at the different current levels between these two strains, as well as the temporal current dynamics, we were able to distinguish these different quinones. For example, for DHNA, lawson, and malodin, we see a much more bigger difference between the NDH2 wild type versus knockout. Whereas for other quinones, we see a smaller difference. And also, over the time, the current increasing rate is different for each of the quinones. So this means that we can harness the NDH2 promiscuity to sense those different quinones and then utilize the different electrical signal pattern to distinguish these different quinone derivatives. And then we may also extend the ability of this quinone sensing to other diverse quinone of interest. And you may find quinones as a plant microbial metabolite, and some of those quinones are manufactured as drugs. And you may also find quinones in the industrial waste. So if we can leverage alpentanol to sense those different quinones, we may be able to develop bioelectronic sensors for drug and pollution monitoring. And we can also use it as a tool to fundamentally understand the plant and microbial metabolism. But before we can achieve this, one remaining question is that what is the mechanism that allow alpentanol to differentiate those different quinone derivatives, even though they share they share a very similar structure. And understanding this mechanism is very important because so we can use the mechanism to link a specific singular pattern to a specific quinone molecule. And so I developed a four-step model to decipher the electron transfer mechanism of NDH2. And in this model, the first step, the quinone have to enter into the cell membrane, and then quinone binds to NDH2. And next, quinone exchange the electrons with the FAD cofactors. And eventually, the reduced quinone will leave the cell membrane and then reduce the external electron acceptors. And I hypothesis that the electron transfer rate for each step is determined by a quinone physical-chemical properties. For example, the quinone entering and leaving the cell membrane is determined by their lipophilicity and hydrophilicity. And quinone also binds to NDH2 with a different affinity, and this will also influence the EET activity. As well as the quinone has a different electron transfer rate with the cofactor FAD, and this will determine if the quinone can pick up the electrons from the NDH2. And then I used a descriptor to quantitatively describe each physical-chemical property. And I used the partition coefficient to describe lipophilicity and hydrophilicity. And I used the autodoc vena to predict the NDH2 and quinone binding affinity. And I also used the bioelectrochemistry to measure the redox activity of the quinones. And then my hypothesis is that an alpentanol acts as a chemical tongue or nose, which use an intrinsic function to filter those quinones for EET. And to this end, I developed a multivariate model by integrating all of those physical-chemical properties and trying to predict the EET activity induced by these different quinones. And from the data at the bottom, you can see that the y-axis is the experimental EET activity, and the x-axis quinone score, which stands for the predicted EET activity. And so there is a correlation between the experiments and the prediction. So this means that we can use this multivariate model to decipher which electrical signals represent which quinone. And in the future, we can use this promiscuous oxidoreductase to sense different quinones and then use the multivariate model to decipher the signal patterns. And I also envision that this framework of using promiscuous enzyme for selective sensing can also be adapted for a variety of other analytes to save the engineering process and allow more analyte to be detected by those microorganisms. All right, so shift the gear a little bit to my second story, where I wish to share a platform that I created for a modular bioelectronic sensing. So microorganisms have naturally evolved many genetic circuits to sense diverse molecules. So making it possible for us to leverage those genetic circuits to sense different analytes using the microorganisms. Well, as I mentioned at the very beginning, researcher in this field tends to use like fluorescence or luminescence as the output of those biosensors. However, these signals have some limitations. For example, they need oxygen to activate, and then detecting those signals need a light source. So if you wish to use those sensors in an opaque environment where there's no light, it will not work. And I was wondering if we can, instead of using fluorescence, using the electrical signals as the output of those biosensors. Electrical signals is very easily to be detected, and it works in very different environment, even in the opaque environment. And we only need some simple electronic components to detect the electrical signals, whereas we have to use some specialized equipment to detect the fluorescence or luminescence. However, even though the conception is very straightforward, using electrical signals as the output of biosensors, but in reality, the engineering process is very difficult because those electron transfer pathways tends to have many different components, and they only works in a specific environmental context. So if we're trying to move those different components from one cell to another one, just like we wish to do with the green fluorescence protein, it will simply don't work. And even though there are some like small devices that we can use to detect the electrical signals, now people tend to use some specialized equipment to measure those electrical signals in the lab, or as those equipment are too bulky and high price for us to use in a practical settings. So I was wondering, can we develop a simpler strategy to engineer bioelectronic sensors and also to measure the electrical signals? So I was inspired by a natural solution to this problem, which is the sender and receiver system. Instead of centralizing all of those signal and transfer components in one cell type, the sender receiver system divide those functions into different cells, so making them highly modular. For example, two examples exist in nature. One is the signaling transfer through the neuron is mediated by the neuron transmitter between a sender and a receiver cells, which allow the signal to pass a very long distance of the neuron network. And the other example is that the bacteria, some of the species can use quorum sensing to propagate the signals in a biofilm or in a community. So quorum sensing send the signals from a sender cells to the receiver cells and activate the gene expression in the receiver cells. And the advantage of this sender receiver system is that one receiver cells can receive many signals from the sender cells so that the signals are intrinsically amplified. And also, because it doesn't need one cells to express all of those components, those cooperation activity allow the signal to propagate through a very long distance. So inspired by this sender and receiver system, I created EcoSense, which is a modular platform to develop bioelectronic sensors. It stands for Electroactive Co-Culture Sensing System, which consists of three components. We have a sender strand that can produce a quinone molecule, and I use a quinone as the signaling molecule to activate the EET in the receiver stream. So as you can see that instead of directly engineering the electron transfer pathways, I use quinone as an intermediate signaling molecule to control the on-off activity of the electron transfer. So let me break down a little bit of each step to explain the mechanism of the EcoSense. The receiver bacteria is a bacteria that can perform EET using a similar mechanism that I have introduced in the first story, which is mediated by the NADH quinone oxidase reductase as well as a quinone molecule. And if I did a phylogenetic analysis, I found that diverse firmicutes, including alpenteron, actually possess an EET locus. However, many of them, just like alpenteron, they cannot synthesize the quinone by themselves, so they rely on exogenous quinones to perform EET. And on the other hand, we have a sender bacteria. So there are many other bacteria that can naturally synthesize DHNA. This is not surprising because DHNA is the precursor of vitamin K2. So many bacteria rely on vitamin K2 monoquinone for respiration. So I also did a phylogenetic analysis and I found that over 2,000 of bacterial species in the database that I was referring to possess the DHNA biosynthesis pathway. And some of the notable chassis that we are familiar with, including Lactococcus lactis or E. coli, which are gram-positive and gram-negative model organisms, respectively, as well as the soil bacteria Bacillus subtilis and the marine bacteria Rubio nitrogen. So this means that we can potentially use those different quinone-producing bacteria in the ecosense system to allow the system to adapt to different environment. For example, the human body or soil or the marine environment. And if we co-culture the sender bacteria and the receiver bacteria, now the sender bacteria can secrete the DHNA into the environment, which can be utilized by the receiver bacteria to perform the quinone-mediated EET. And we can further engineer the quinone biosynthesis pathway to make it inducible. So specifically, we knock out the MAN-A to stop the reaction at DHNA because DHNA is the molecule that is used for EET. And we also knock out MAN-D and control its expression on a plasmid under an inducible promoter. So that we can induce the quinone biosynthesis with a environmental input. So you may ask why I specifically engineer MAN-D. Well, there are other 6 enzymes in this pathway. And I also had this question a couple years ago. And what I was realizing was that we lack a deep understanding of the quinone biosynthesis pathway for us to determine which enzyme is regulable on a plasmid. So to answer this question, I reached out to Oliver Goshin and Jiang Guo at Rice University, where we used a systems biology approach to understand the regulatory mechanism of the DHNA. So what we found was a multi-layer mechanism that can regulate DHNA biosynthesis. It is regulated by a reversible flask at the MAN-F, which is the first enzyme, and also allosteric feedback inhibition of MAN-D from the end product DHNA. So we did some mathematical modeling and also used the computation simulation to predict the quinone, the DHNA concentration, showing you in different color here as a function of the MAN-D or MAN-F expression level. So what we found was that only when we change the concentration expression level of MAN-D, we can see a differential DHNA concentration. Whereas if we move the MAN-F concentration, increase the MAN-F concentration, we cannot see the increase of the DHNA. So the take-home message here is that MAN-D is the key regulatory enzyme in the DHNA biosynthesis pathway, and we can use to regulate the quinone biosynthesis. And building on all of this previous understanding, I created 2 co-cultures for ecosense. So I co-cultured alpenteron with Lactococcus lactis, which is a model gram-positive organism that is also found in the human gut microbiome. And I also co-cultured alpenteron with E. coli, which is a model gram-negative organism that has many different genetic tools that we can use to engineer the cell for biosensing. And in this data, you can see that we test the co-culture in the bioelectrochemical cells, and we measure the current density here. And we observe no current if there's only one strain, or if one of the strain in this co-culture is dysfunctioned. And only when we co-culture the 2 strains and both of them are functional, we can observe a current generation. And to enable the co-culture to detect specific molecules, I then control the quinone biosynthesis using inducible genetic circuits. So I use the lysine inducible genetic circuits to control the Mendel expression in Lactococcus lactis. Lysine is a antimicrobial peptide here. And also I use QMate inducible circuits to control the Mendel in E. coli. And QMate is a small molecule that we often use for biosensing demonstration. And in both cases, you can see that when we add in increasing concentrations of the respective inducer into the co-culture, the co-culture generates a increasing level of the current in response to the inducer. So which means that EcoSense system can quantitatively respond to these different analytes. And as I mentioned that one of the beauty of this system is modularity. We can change different center strain and also the associated genetic circuits to enable the system to detect the different analytes and also in different environments. And to demonstrate this, I constructed 3 additional sensors to sense health and environmental relevant analytes. So I further engineered L-lactis to sense an anhydrous tetracycline, which is a derivative of the antibiotic tetracycline. And I used the sensor to sense ATC in the milk sample. And I also engineered E. coli to sense arsenate, which is a heavy metal contamination. And I used the sensor to sense arsenate in the bayou water. And I also engineered E. coli to sense hydrogen peroxide, and which is a inflammation biomarker people use in the gut or in the oral cavity. And I use the sensor to detect the hydrogen peroxide in the artificial saliva sample. And in all of these 3 cases, even though these involvement are very complex with many undefined molecules, the EcoSense still remains functional and responsive. And when we add the different inducer into the respective involvement, we can see an increase in production of the current from the core culture. And we also demonstrate that EcoSense functions in complex gut microbiome. And this was a collaboration with Robert Britton and Duolong Zhu from the Baylor College of Medicine. So we connected 3 human gut microbiome sample from 3 human daughters, and we tested the L-lactis and L-pentanol co-culture to see if they can sense lysine in the microbiome. And L-lactis and L-pentanol are natively reside in the gut microbiome. And despite that, there are some background from the native gut microbiome. We can still see that in all of the 3 samples, the co-culture generates a higher current in response to the lysine concentration. So this means that EcoSense also functions in a complex microbial community and showing the potential for use for in vivo applications in the future. And lastly, I also partnered with the collaborators from Tufts, Samir Sonkuseli and Kudan Saha, to trying to make the sensing system more accessible to different people. So as I mentioned that traditionally people rely on some specialized equipment to measure electrical signals, and this is too bulky and expensive to use in field applications. So we instead engineered a miniature device to allow locals and portable sensing. So this device is made based on the microbial cell conception and is the size of a coin. And we can even use some simple multimeter to detect the voltage or current signal from the device. And we were able to use this small device to detect the ATC from the milk sample. So this showing you that in the future, we may use EcoSense as well as this small device along with a accessible commercialized multimeter to perform bioelectronic sensing at different on-site settings. For example, even at our home or for wastewater detection on site. And also, I'm collaborating with Rafael Verduzco and Xin Yuan at Rice and trying to develop a hydrogel encapsulation method for those microbes. In the real application, we don't want those cells to be released from the detection zone into the environment because those cells are engineered. So we're trying to use hydrogel to encapsulate those bacteria, but the problem is that the hydrogels are not conductive. So Xin Yuan created a quinone grafted chitosan. So she grafted the one form of the quinone motif to the backbone of the chitosan, and we used this hydrogel to encapsulate alpenterol. So this TEM image connected by Xin Yuan, you can see that when there's no hydrogel, the alpenterol was barely attached to the electrode. Whereas if we encapsulate the cells, the alpenterol are highly enriched around the electrode. So we wish that with the hydrogel, we can increase the electrical signal generation from those microbes and also prevent them release into the environment. So with that, I would like to conclude my talk today. So I have shown you that microbial bioelectronic sensors are promising techniques for environmental and health monitoring. And we harnessed the promiscuous oxidation reductase in alpenterol for selective sensing of quinone derivatives. And I also created a electroactive core culture sensing system, EcoSense, that allows modular bioelectronic sensing in distinct environment. And with that, I'd like to thank my lab, my PhD advisor, Caroline Alcoff Franklin, and all of other lab members from the CAF lab for their support. As well as I would like to thank my collaborators and the founding agency, especially I was also funded by the NSF Research Chairmanship Fellowship at Rice University to enable some of my work here. With that, I will be happy to answer any of your questions. Thank you so much.
Thank you. This is exciting. Let me check whether Dr. Nasser has any comment or questions.
Thank you, Siliang. It's fantastic work. Congratulations on that. I just have a general question. In in the field, we are either aspiring to scale up or scale out these applications. And in your case, probably it's more likely to scale out. I'm borrowing terms from computer science and electronics. When do you think we will be able to scale out these proof of concept applications that you just presented to us today?
So yeah, I think that's a very good question. And everyone here wish to push the synthetic biology to the field applications at some point. So I think in our case, those microbial bioelectronic sensors, I think they are ready for like field applications. As I demonstrate that we can use some simple devices to sense the signals, and also the bacteria that we are using were considered as safe by FAD. And so yeah, so I think that as long as we figure out how we deal with those engineered microbes and trying to use the hydrogel to prevent them leaking to the environment, we were able to use those sensors in the field.
Thank you.
Do you have any other question or comment?
No, thanks for that. That's great.
Thank you. So let me ask one question. So I like the second part the most. This is the really amazing. But my question is about the first part. So you show you know basically the promiscuous sensing and then saying this is a sort of specific depending on the pattern of the output, right? If I understand the correctly. But I probably missed that part, but if you change the concentration of individual molecule, I also believe the pattern also will change. And then try to understand how you can differentiate between different compounds sensing. That part was not clear to me.
This is a very good question. So far, the data I'm showing you here, there's only one concentration for
Right.
each of the different quinones. And in terms of how we going to differentiate the signals for under different concentrations, I think that we need a more advanced model
Mm-hmm.
to decipher the signal patterns. I have been using linear regression
Mm-hmm.
for my prediction, but
Mm-hmm.
I think this will become constrained or limited if we trying to move towards quantitative sensing of different molecules.
Got
And I
it.
think my colleagues now are developing a more advanced model based on machine learning.
Mm-hmm.
And then we hope that using the machine learning model, we will be able to differentiate those different signal patterns for different concentrations
Got it.
for selective sensing.
And then can you also comment on the response time of your sensor in general? Because I see, I mean, some time scale, I mean, in the later slide, but I probably missed the earlier slide. What is time scale of the response?
So this really depends on what is the mechanism of the sensing, right?
Mhm.
So in the first case, in the first story, we use the quinone and the oxidoreductive bonding for
Mhm.
signal generation, and this doesn't uh, rely on transcription or translation. So
That's
it's
very
very
fast,
fast.
right? Yeah,
We can detect the signals within like couple seconds
that's
or like
great.
5 minutes, say.
Hmm.
Um, the second story, I was using genetic circuits
Right.
for the signal generation, and this relies on transcription and translation. And this typically is slower than the post-translational
Right.
response. So you can see that the response time was ranged from half an hour to couple hours.
Sure.
Uh, so there are some like rooms for to improve those response time. For
Hmm.
example, some of our colleagues already engineered the electron transfer protein as a protein switch,
Hmm.
so that the protein switch can bind to a analyze and then become function within
Hmm.
like 5 minutes. So yeah, so I think the response time is really depends on like what's the mechanism of the sensing, and we can tailor for like different applications in terms of what you want for your analyze to be detected in what was the time frame that you want.
So that is my impression, but I need to confirm. I mean, another way I can say, life is fair or challenging because you have the very great speed in the first sensor system, but you also have some limitation in terms of specificity. Second one has more specificity, but the speed is slow. So it's kind of very difficult to balance or making everything good. So that is kind of our challenge we need to solve, I guess. So, but still very fantastic. I love the your talk. Let me check whether we have the question from audience. I do not see. Okay, and then I see also the 1 minute after 11 a.m. Central Time. So probably time to close. So thank you everybody for joining and staying today. We'll meet again on September 4, Thursday, the same time, the same Zoom link. We'll have Dr. Tuba Fur at National Institute on Biomedical and Biomedical Imaging and Bioengineering of National Institutes of Health, and another, program manager. Also Dr. Shohei Kitano at National University of Singapore. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again, and I stop recording.